Hello everyone and welcome to today's webinar entitled Rapid Biotherapeutic Charge Variant Analysis by CZE-UV and CZE-MS. So I'm Jim Thorne of SCIEX and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and is brought to you by us in SCIEX. To learn more you can visit us at sciex.com. We'd encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You can also submit any technical issues here as well if you have trouble hearing or seeing the presentation. So I'd like to now welcome our speakers, Dr. Christian Baumann, Accurate Mass Workflow Specialist, and Dr. Whitney Smith, Field Applications Specialist. Dr. Smith, you may now begin your presentation.
My name is Whitney Smith. I'm an application specialist at SCIEX for CE and CEMS. And I just want to give you kind of a little bit of info on what we can do to help you with characterization of your monoclonal antibodies. It can be quite a challenging endeavor, and we have two really quite simple methods, technologies, if you will, that can help you in this type of analysis. So why is a charge variant analysis something that is important for the pharmaceutical industry? Well, regulatory agencies require analysis of the charge variants, and they say that everything must be characterized. But this is not the only reason why we need to characterize these things. Some of the critical quality attributes, or CQAs, can induce surface charge changes to the molecules, and this can then affect stability, efficacy, and safety to the patients that we're trying to help out with these drugs. What does SCIEX offer to analyze charge variants? Well, as you see on the left, is the P800 Plus with our CZE kit, and on the right we have the zip chip with the SCIEX 6600 mass spectrometer. We can utilize the zip chip with the zip chip charge variant TOF kit, and this allows us to identify each of the post-translational modifications, or PTMs, in the product. Once these are identified, we can then use the P800 Plus with the CZE kit to do routine analysis of the product itself. As you see here, the CZE kit is on the bottom. It's 3 components. It comes with pre-mixed chemicals and a pre-assembled capillary. And it is basically plug and play on the P800 Plus. We have methods on the website which you can download, and it's very easy to use, very simple sample preparation. And we're going to look at some comparisons here in just 1 second. So here you see on the top row, you see 4 different monoclonal antibodies: trastuzumab, adalimumab, the Nist Map, and infliximab. That is the CZE electropherogram that you see on the top row. And in comparison, the zip chip charge variant profile is on the bottom, and they are nearly identical, which allows quite easy comparison. So here's a more detailed look at the profiles. Some of you may be familiar with capillary isoelectric focusing, which is a detailed analysis of the product and shows the acidic and basic variants, as well as determines the isoelectric point. And how does that compare to the CZE? Well, it's basically a mirror image. In CIF, the acidic modifications come out before the main peak and the basics come after, but with CZE, the basic variants elute first, and then the acidic is following. Here is an image of what the zip chip system looks like. It is an easy interface that you attach to any mass spec. Here it is attached to a 6600. It's a chip-based separation. It comes with a kit, and you prepare your sample and control it via the mass spec. So why do we want to combine it with the 6600? Well, it offers high sensitivity. It is ultra fast, and we are able to collect high resolution MS data. We can have an enhanced quantitation with linear dynamic range, and it is extremely easy to connect with the zip chip. Our software allows for quick data processing. What are the benefits of the SCIEX workflow? The CZE MS kit offers charge variant analysis in the native state. The subunit level analysis provides domain-specific modification information. Peak identification is easily achieved through the high-resolution native chip for the zip chip. And the platform is basically it's kit-based and is very simple to install and run. So let's look back at the infliximab that we saw previously. Again, on the left, you have the CIEF profile with the acidic variants on the left and the basic variants on the right. Now, in the middle, you have the CZE profile with the basic variants on the left and the acidic variants on the right. And as you can see from the third picture on the right, the basic variants are on the left side and the acidic variants are on the right. So it is one-to-one comparison with the CZE profile. Now let's look at this in more detail. So we have three main peaks for infliximab, and we're going to look at what these variants actually are. So we look at our zip chip profile. Peak 1 is a double lysine variant, peak 2 is a single lysine variant, and peak 3 is the main peak. And you can see by the change in the mass. Another example, a less complex example, is the Nist Map. It has one acidic variant, one main basic variant, and then the main peak itself. And as you can see, is similar to the infliximab. The CIEF and CZE profiles are mirror images, and the zip chip profile is the same as the CZE profile, which makes monitoring very easy. This is just a closer look at the zip chip profile to identify the peaks. Peak 1 would be that basic variant, peak 2 is the main peak, and peak 3 is the acidic variant. And what are those peaks? Peak 1 is the lysine variant, peak 2 is the main peak, and peak 3 has a mass shift of 6 Daltons, which is most likely due to deamidation. So we wanted to prove that this workflow was good at doing charge variant characterization. As we mentioned previously, we utilized two workflows for identity and monitoring workflow. One is the identity, and that is with zip chip with the charge variant TOF kit on our triple TOF 6600. And our monitoring workflow is the P800 plus CE instrumentation with CZE rapid charge variant analysis kit and the pre-assembled cartridge, which makes it a nice plug-and-play system. The molecule we used to prove that these two workflows are well suited to each other was Humira, was diluted to 1 milligram per mil to represent stress conditions. It was incubated and Ides digested at 40 degrees for 10 days. To prove the portability of the assay from identity to monitoring, we replicated conditions on both separation tools of the zip chip and the P800 Plus. As mentioned on the previous slide, to further improve separation and characterization, we did an Ides digestion. This cleaves the molecule at the hinge region and results in a large FAB region and the FC region, as seen in this slide here. The Ides cleaves the linkage between the two regions and at the hinge region. And what you can see here is we have the main peak and the FAB2 and the FC regions and the acidic variance for each of those peaks. So for the FAB main and then the FAB acidic, we have a +3 Dalton, which is most likely deamidization. Then we have the FC basic, which is a double lysine variant. The FC basic, the second FC basic, which is the single lysine variant. And then we have another acidic peak, and then the main FC peak, and then 2 more acidic variants. And as you can see, the profile of the inset is very similar to the profile identified with the zip chip. To confirm that the molecule is in its native state, we were able to separate the 2 FC2 fragments generated from the Ides enzyme. We confirmed the method-induced artifacts were not present due to the enzyme digestion, and that we did analyze this molecule in a nearest native state. With the Ides cut, we can see the majority of the molecule has been cut, as we see primarily the FC region, which is in good, and we see minimal dissociation into FC2, as you can see in the bottom part. In quick summary, both of the analyses are only about 10 minutes for domain-specific charge variant profiling and identification. Both systems use ready-to-use kits, which are easy to operate and install. The zip chip is capable of analyzing intact molecules and doing the subunit level analysis. And there is a one-to-one comparison between the CZE with UV detection, which would be our CZE kit on the P800 Plus, and the peak identification using CZE on the MS, with MS detection. And now I will hand it over to my colleague Christian to talk about more capabilities for zip chip.
So hello everybody, my name is Christian Baumann. Thanks Whitney for giving this really nice insight in charge set analysis using the zip chip MS coupling. I will guide you through some other applications for zip chip MS. You heard about the intact antibody characterization, like the charge state variant analysis, but also of course you can just monitor the intact mass of an intact antibody and also process it through our software. Plus you can also handle more complex samples like peptide mapping samples. So as you can see, with a runtime as short as 10 minutes, we can obtain close to 100% sequence coverage with the zip chip MS coupling. We are well able to separate on a chromatographic or electroferrographic basis modifications or modified peptides like glycopeptides, deamidated peptides, from their native peptides. And also we were able to detect additional peptides in the peptide mapping that we couldn't see with LCMS, for example, very small or very large peptides. I will show examples in a minute. Plus, we can also analyze the cell culture media where the antibodies are grown in. It's very important there to monitor the nutrients like amino acids, sugars, and other ingredients in the cell culture media during the process. Because the instrument is so fast, we can do quantitation on these very sharp peaks and very narrow peaks, as well in TOF-MS, so just to monitor the peptide mass, but also run real MRM quantification on the peptides. The quantitative performance of the SIP-chip MS system is really similar to the gold standard, the LCMS, when it comes to quantitation, in terms of sensitivity, in terms of complexity, also in terms of dynamic range. And last but not least, the fast run times. I cannot stress it enough. In 4 minutes, you can analyze a whole bunch of cell media components. I will show some examples in the next slides. But first, I want to come back to the slide that Whitney already showed, because for these more complex samples, like peptide mapping or like cell culture media analysis, we really need this instrument to be very sensitive. We need this instrument to be very fast, so we can run up to 100 Hertz MS. And for quantitation, of course, we will need an excellent linear dynamic range, which in this instrument is 5 to 6 orders of magnitude. And last but not least, it's really easy to connect with the SIP-chip box, like Whitney showed before. Whitney also mentioned the Biopharma View and Biopharma View Flex software suites, which we use to process the data. Biopharma View can do an automatic deconvolution of the data, process intact masses, and also show the sequence coverage for peptide mappings, for example. So on this slide, you see just an example of a peptide mapping run, a SIP-chip, together with the MS. You can see the runtime is only 10 minutes, and you see very narrow, very sharp peaks, very good separation. And what you can also see is that we almost reached a sequence coverage of 100% for the peptide mapping. On the next slides, I just show you some examples for the very good separation of modified peptides. For example, in this case, we see a very good separation of the deamidated peptides versus the non-deamidated peptides, which is not always given in LCMS. This is so important because the mass of a non-deamidated and a deamidated peptide only differs by 1 Dalton. So you will have overlapping isotope patterns. So it's very crucial here that we can get a baseline separation in order to quantify the modifications on the peptides. So next slide. The same holds true for glycosylated peptides. So also here we need a very good separation to be able to quantify relatively the glycosylated peptides from the non-glycosylated peptides, and also do a relative quantitation between the glycosylation sites. Also here you can see there's almost a baseline separation between the glycopeptides. This is also not always given in LCMS. Another example, which I mentioned earlier briefly, some peptides we miss in LCMS. For example, very small hydrophilic peptides, they tend to elute very early in the injection peak. So you don't get a really clean spectrum in LCMS sometimes. The same holds true for the very big peptides, the very big hydrophobic ones. They usually elute quite late in the waste fraction of the LCMS run, and therefore also there we can't get very clear spectra here. Due to the orthogonal separation technique, like SIP-chip or CE in general, we were able to detect those peptides, which are challenging in LCMS. Now I'm coming to the cell culture media analysis. So as I said earlier, it's crucial during the process to monitor the nutrients and the ingredients of the cell culture media in order to make the cells grow and produce your product. In this example, you can see a very nice linearity. So we did just a calibration curve and spiked in amino acids standards. So you can see it's very linear, it's very reproducible. Each point is a triplicate. So this is really a quantitation you would expect from a triple quad instrument or from LCMS analysis, which is the gold standard for this kind of analysis. And on the next slide, you can see a direct comparison. So in the green bars you can see here, I just took an excerpt of the ingredients we monitored here. The green bars on the left of each amino acid are the actual values. So we used a commercially available cell culture medium here. And in gray, you can see the gold standard. This is LCMS quantitation. And the two blue bars are, the light blue one is the zip chip based quantitation with the same MS instrument, but just based on the peptide mass. And the dark blue bar is the same quantitation on zip chip, but using the MRM transition, so based on the fragments. And you can see all the quantitational results here are in the same ballpark. It's really reproducible and it's really comparable to the gold standard we were using so far. Plus, it's only 4-minute analysis. So with this, I just wanted to point your attention a little bit to other applications we could use the zip chip for. And I hope you enjoyed the presentation, and we are open for questions now.
So thank you very much, Dr. Baumann and Dr. Smith, for a very informative presentation. We'll now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so. You'll need to click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So the first question then, with CZE UV, do you use bare fused silica or coated capillary? And if coated, what kind of coat? Can you comment on the buffer system and the stability of the capillary, please?
So thanks for the question. The CZE kit uses a bare fused silica capillary. You can, although you can use a neutral coated capillary if you would like. Typical results, if the stability of the capillary, meaning I assume you mean how many runs you can get out. We do see some coating at the higher end of injections, for number of injections, and then but we do have a procedure to recover the capillary, and it works very well. So,
thank you, Whitney. The next question, in CZE, you can normally not speak about basic or acidic peaks. Why is this mentioned in the kit? Because all peaks will have a positive charge.
It's because the variants are in relation to the PI of the main peak, are either a shift in the basic region towards the basic pH or towards a more acidic pH relative to the PI of the main peak. Thank you.
The next question, can you say something about the dynamic range for quantitative workflows compared to LCMS?
Yes, I will take that one. It's about the dynamic range. Was it that question?
Yeah, so can you say something about the dynamic range for quantitative workflows compared to LCMS?
Yeah, with this project, it's quite new. We haven't fully fully elaborated the whole dynamic range. But in general, the instrument has a dynamic range of 5 to 6 orders of magnitude. For this kind of analysis, we didn't need that because we didn't monitor all the kind of ingredients in there. But I guess we will go in the same direction. There's more to be tested here.
Great, thanks, Christian. The next question, is the sensitivity of ZipChip MS comparable to LCMS in terms of targeted quantitation? I guess that's a related question then.
That's really related to this one, yeah. I can say yes. The difference is that the concentration for the ZipChip samples have to be a little bit higher because you can only inject a few nanoliters in the system. But if you look at the overall amount you put on the column or on the capillary here, it's really comparable. Plus, because we run at so low flow rates, I think, Whitney, correct me, it's 30 to 50 nanoliter per minute or so. We barely see, or we see CESI typically, we don't see any kind of suppression effects from the matrix, from the background, from other analytes. So I would say yes, it's very comparable, plus very much faster.
Great, thank you. Then the next question is a simple one. Is the ZipChip 96 well plate compatible?
Yes, it is.
There we go. Quick answer. Thank you. Next question. How quickly can you generate a peptide map for a typical antibody sample? And what concentration of sample is needed for a peptide map?
Well, it depends on if you're wanting high resolution or high speed. Typically with ZipChip, the high resolution chips take about 10 to 15 minutes. We start with a concentration of 1 milligram per mil, but we can also go lower than that. But at the high speed chips are around 7 minutes.
And that's also comparable to what we use in LCMS typically. So you don't need to make up two different samples here. You can use the same sample for LCMS and ZipChip MS if you want to do both.
Very good. Thank you. The next question, let's see, that's related as well. Let's move on to this one. Is it possible to use the ZipChip for nucleic acid analysis and interactions between proteins and nucleic acids?
I am honestly not sure. I have not looked into that, but I will get back to you with an answer on that.
That's great. Thanks, Whitney. All right. And then maybe one last question, maybe related to one we had before. How much sample is needed for each method?
For the CZE UV kit, you need, again, 1 milligram per milliliter of sample concentration, but you can use as little as 5 microliters because we have nanovials for the P800 plus. So 5 micrograms for the CZE UV and about 50 microliters for the ZipChip, depending on what kind of vials you use.
Great, thank you. So I would say we should. We've run to the end of the allotted time here. So thank you very much to Christian Baumann and Whitney Smith. Do you have any final comments for our audience before we wrap up?
Just thank you very, very much for joining the webinar. And if you have any questions, feel free.
Great, thank you. Well, thanks again, both of you, for your time today and for the important work that you've presented here. We'd also like to thank LabRoots and SCIEX for underwriting today's educational event. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. If there are any questions we didn't have time for, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be reviewed on demand LabRoots will alert you via email when it's available for replay. I would encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.